Hello, my name is Dr. Matt Piercy. I'm a faculty member at Yavapai College. I'm making a series of videos that deal with the biology and the current treatments that are in progress for the COVID-19 virus. So the subject of today's video is going to be a vaccine that's currently in development. It's in clinical trials and has been for a few weeks now. So what I'm going to do is talk about how this vaccine actually functions, the concept behind how it's made, how it's administered, and how hopefully it will work. So if you would like more information about this particular topic, you can either Google mRNA 1273, which is the actual name of this vaccine, or you can go to the link shown at the bottom of this slide. It has further information, and some of those links are actually where I got some of these figures from. So to start this out, if you look at the left-hand side of the slide, what you see is a figure of the COVID-19 virus. It shows a bunch of different parts of it. There are two parts of the virus that we're going to be concerned with today. The first are the spike proteins. Those are the proteins that are on the outside of the virus, and you can see those encircling the virus. The job of the spike protein is to function essentially as a key. It's what gets the COVID-19 virus into our cells. So if you think about it, if we could cover up or prevent the function of the spike proteins, then we could prevent the virus from infecting our cells. And that's the whole goal of this particular vaccination. So if you look also at the middle part of the figure, you'll see something labeled RNA. That's that red material in the center. The RNA is basically the genome of the virus. What that means is it's the set of instructions on how to build new viruses. So remember that viruses aren't alive. So the only way that the virus can replicate itself or make new copies is to get inside of our cells. Once a virus is inside of our cells, then its genome is used as a set of instructions by our cellular machinery to make new copies of the virus. So here's what we're going to do in this vaccine. We're going to teach the body how to destroy the spike proteins without ever infecting the person with an actual COVID-19 virus. So here's how we're going to do it. Everything on this slide is happening in a laboratory. This is not going to be injected into anyone. We're not putting live COVID-19 into any patient to try to provoke an immune response. So if you look in the middle of the slide there, it says RNA for spike protein. So if you look back at the figure on the left where it's labeled RNA, that's the set of instructions on how you build the entire virus. Somewhere in there is a short section of that RNA that has the instructions on how you build just the spike protein. So what we're going to do in a lab is take the virus and take out just the RNA for the spike protein. That's it, right? So the next thing we want to do is take that RNA for the spike protein and we're going to surround it with some molecules that make it what we call lipid soluble. What that really means is that we're going to enable that RNA for the spike protein to dissolve into our cells. That's all that really means. So if you look at the top right-hand side of the slide, what you see is essentially the vaccine. It consists of two things. One, we have the RNA, which is just the set of instructions on how to build the spike protein, and that's it. There are no other parts of the virus involved. And that RNA with the set of instructions on how to make the spike protein is surrounded by molecules that are just going to enable that RNA to dissolve into our cells. So that's essentially what the vaccine is going to be. So if you look at this slide at the top left, this is just a human cell. So this could be just any generic human cell. If you look at it, it's completely surrounded by all these different shapes. And those shapes are just meant to represent all the molecules on the surface of our cells. They have different shapes. Some look like stop signs, as you can see there, some like triangles, so on and so forth. That's essentially accurate. There's a bunch of molecules on the surface of our cells that have different three-dimensional shapes. And here's the thing. Our immune system knows each and every one of those shapes. They are what are considered self. Our immune system knows to leave every one of those molecules alone and therefore the cells alone because those are ours. They belong to us. They're not a foreign microbe. So if you move to the right-hand side, what I'm trying to show you there is how we administer the vaccine. So this is just the person, right? Although the person is just one cell in this picture. And then we're going to inject them with the vaccine that we described on the last slide. So after we've given the person the vaccine, The RNA that has the set of instructions on how to build just the spike protein is now inside of one of our cells. So we're composed of literally trillions of cells. And so this vaccine is going to be given to us, and it's going to result in hundreds, maybe a few thousand of our cells having the RNA for this spike protein inside of them. It's not enough of our cells to really matter. But here's what's going to happen once that RNA for the spike protein gets inside of our cells. That RNA looks exactly like our RNA, like human RNA. So our cell considers this a set of instructions that were proposed or built by our cells. So in other words, that RNA for the virus spike protein looks exactly like our RNA. So what our cell is going to do is what it always does. It's going to use that RNA as a set of instructions to build a protein. But what's going to happen is our cell is going to build COVID-19 spike proteins. So if you look on the right-hand side of the slide, you see the end result of that. This is one of our cells. It's exactly the same as the ones we've been talking about, except our cells now have the COVID-19 spike protein on them. That's the only part of the virus that is inside of this person's body. It's just the spike protein. The rest of the virus isn't there. So the person isn't infected with the virus. The person is not going to get sick. They're not going to get ill in any way, shape, or form. But they do have a few of their cells that are now expressing or have the COVID-19 spike protein on their surface. Now remember when we talked about that picture on the left, we had all those molecules normally on the surface of our cells, but they have three-dimensional shapes that our immune system knows are ours. So the immune system leaves that cell alone. Well, that's all going to change now that our cell has the virus spike protein on its surface. That is foreign. That three-dimensional shape is not part of our body, and our immune system knows it. So this is going to start a process that's called provoking an immune response. Simply put, it just means that our immune system is going to see that cell with the COVID-19 spike proteins on it, and it's going to think it's foreign. It's going to consider that cell the same way it would any other microbe. And so what it's going to do is mount an immune response to it, which is going to result in that cell being killed. So we're going to lose a few thousand cells, but the process that our immune system is going to use to kill that cell is going to result in the production of what we call neutralizing antibodies. And so those antibodies are shown on the top right of the slide. So those antibodies are going to have the ability to bind to that spike protein. So at the end of this vaccination process, it's going to take a couple weeks. We've lost maybe a few hundred, maybe a few thousand cells. That's not really a big deal. But what we've done is shown our immune system the COVID-19 spike protein. Our immune system has seen it, recognized that it's foreign, mounted an immune response to it, and so at the end of it all, we're left with antibodies in our body that bind to the COVID-19 virus, which, by the way, we've never been infected with. But if we are infected with it, our antibodies are already there waiting for the virus. So later on, if we were to happen to be infected with the actual COVID-19 virus shown here, our body already has the antibodies made that bind to the COVID-19 spike protein. So the antibodies will do their job. They'll do what antibodies do. The antibodies will bind to the spike protein. That will prevent the spike protein from being able to bind to our cells. That will prevent the infection of our cells by the COVID-19 virus, and we most likely will not get ill at all. So that's how that vaccine is going to function. And it's in the beginning stages of clinical trials, and we'll see what develops with this vaccine in the coming weeks and months.